Good afternoon, and welcome to today's webinar sponsored by the North American Vascular Biology Organization. I'm Linda Shapiro from the University of Connecticut and a member of the NAVBO Education Committee. I will be moderating today's session. We are quite pleased to welcome our speaker, Bill Sessa from Yale University, who will present his work entitled Lipid Metabolism by the Endothelium. I'd like to take this opportunity to congratulate Dr. Sessa on being named the 2019 Earl P. Bendit Award, which is NAVBO's highest honor. Congrats. I also want to welcome Ana Gamez-Mendez, also from Yale. She will monitor today's questions. The questions will be handled in two ways. Throughout the presentation, you can type your questions into the question box on the control panel. These questions will be answered at the end of the presentation. Dr. Gamez-Mendez will compile and then pose the questions to Dr. Sessa. At the end of the question and answer period, provided there's time, attendees will be able to ask any additional questions. If you have a question, please raise your hand by clicking on the hand icon on the left side of your control panel. You'll be recognized and your mic will be unmuted. You'll then be able to ask your question live. Before we get started, I want to go over some logistical aspects. Throughout this webinar, you're able to switch between the phone audio and computer audio in case you're having a problem. You can see this information on the audio section of your GoToWebinar control panel. If you experience a few words being skipped in the audio, it may be your Wi-Fi connection. Connecting to the internet through a hardwire should remedy the audio. If you experience any technical problems, please click the Help tab at the top of the control panel. Scroll to the bottom and the help screen for the technical support phone number. This webinar is being recorded and archived on the NAVBO website for future use, and relevant papers are posted on the handouts section of your control panel with links to these in the chat section. Dr. Sessa's principal contributions to science revolve around the molecular aspects of endothelial nitric oxide synthase activation and how eNOS regulates angiogenesis, vascular permeability, atherosclerosis, and vascular modeling. This webinar, Lipid Metabolism by the Endothelium, will be closely related to his talk, New Insights into Vascular Control of Lipid Metabolism, that he presented at the intersection of lipid metabolism and vascular health session at Vascular Biology 2018. If you missed the meeting, now is your chance to hear Dr. Sessa's presentation. And if you were at the meeting, this is your chance to hear a more in-depth talk and ask questions you weren't able to at the meeting. Welcome, Dr. Sessa.
Thank you, Linda. Thank you very much for the invitation. I'd like to thank those of you who decided to call in for this. So I want to take you through sort of the logic of why we got involved in this area. And this is work largely driven by Andrew Kuo, who was a graduate student in the lab at the time, who's now a postdoc in Tim Law's lab up at Harvard. So the question that we were interested in was, how does the endothelium process triglyceride-rich lipoproteins and fatty acids? There's a well-characterized model that was largely based on pioneering work by several investigators, including Stephen Young, who came up with a model by which circulating triglyceride-rich lipoproteins, as well as fatty acids, particularly the TG-rich lipoproteins, will dock onto the protein lipoprotein lipase, which is also docked onto a receptor or a binding protein called GPI-HBP1. And it's the docking of LPL to this particular dimer that allows for the lipolysis of lipids from the triglycerides and the transfer of fatty acids into the endothelium. And there's good data that one particular receptor for fatty acid or uptake mechanism is through CD36. And then those fatty acids that are released upon lipolysis of the lipoproteins will then be transported across the endothelium to be taken up by adipose tissue or to be used by striated muscle, particularly skeletal muscle or cardiac muscle, for energy. And so this is a well-characterized model. There's human genetics based on this. And this is the major way by which adipose-derived fatty acids get processed and stored and released into tissue for energy. But the question that we had in the lab was, do large vessel endothelial cells process fatty acids or lipoproteins? Because when you think about atherosclerosis, it's a large vessel disease for the most part that occurs in bifurcations of vessels. So So the question is, this takes place in the microcirculation, but how do the large vessels handle this, number one? And then the other sort of conundrum is that GPI-HBP1 is a uniquely expressed GPI-linked protein that's only expressed in capillary endothelium. So it was unlikely that this pathway that is described on the slide could explain what was happening in large blood vessels. So with that background in mind, we became interested in this question. The other thing that was sort of coming out at the time was there was good human genetics suggesting that triglycerides were a risk factor for heart disease, independent of cholesterol, diet, and smoking and things such as that. And so the model is that, again, you have a triglyceride-rich lipoprotein in the circulation. It can be metabolized by lipoprotein lipase that's docked on GPA-HBP1. And that we knew that lipoprotein lipase was also negatively regulated by ApoC3, angiopoietin-like 4, or positively activated by ApoA5. And through genetic analysis of patients that have less or more risk, it was shown that any mutation that reduced LPL activity would result in the increase in triglycerides in plasma and a higher risk of coronary artery disease. And conversely, if you had a gain of function of LPL activity or a loss of function of the two negative regulators, you'd have a decreased triglycerides in blood and decreased risk of coronary disease. So again, you're talking about plasma lipids correlating with coronary disease, which we believe really must affect the large vessel endothelium in the heart, not necessarily just be a phenomenon taking place in the capillaries. And if you think about this in the context of what we know, this triglyceride component seems to be very important for cardiovascular risk. So when you look at the literature in terms of the effects of statins in reducing mortality and reducing risk, throughout the last 20 years or so, since statins have been introduced, it's been shown through aggregate analysis of multiple epidemiologic studies as well as clinical trials that there's about a 35%, 25 to 35% aggregate risk reduction due to statins, which is amazing if you think about billions of people being treated with these drugs. But we know that there's more risk to be treated because, again, there's a certain group of people that do not respond to statins. And also, when you look at the absolute risk, it takes about 60 patients to be treated for one patient to have a benefit effect. So there's been additional trials trying other ways of augmenting the effects of statins, and we know a lot of these already. So if you lower cholesterol using independent approaches such as PCSK9 inhibitors, that will reduce risk by an additional 15%. So that means you can get to levels of about 40 milligrams per deciliter by PCSK9 inhibitors, and you get only an increase of 15% risk reduction on top of what statins give you. Also, there was the highly touted trial of Cantos trial, where they neutralized IL-1 beta on top of the maximally tolerated dose of a statin, and that reduced risk by about an additional 15%. And this was really an important trial because it was really the first demonstration of a drug that didn't target lipids per se that had an impact on risk reduction. So that was a highlighted trial that was very important for the field. But more recently, actually, there was a trial with omega-3 fatty acids, which is called DHA or EPA combos, and these reduce triglycerides. And there was a trial several years ago called the ASCEND trial. This only reduced risk by 3%. But trials that came out late last year showed that ultra-pure EPA from the company called Amarin reduced risk by about 19 to 25%, which really shocked the entire field because what this shows you is reducing triglycerides on top of a maximally tolerated dose of statins had a tremendous improvement of risk reduction. So this really argues that understanding how lipids are processed by cells in the body is very, very important. Okay, so why did we actually end up doing what we did? Well, there's been data in the literature that's been published already showing that if you look at electron micrographs of animals that are on high-fat diets, whether it be a mouse or a rabbit, that you can see these inclusions of lipid droplets in the endothelium using electron microscopy. And this over here, you can see it's an opaque circle, which is a delipidated lipid droplet. And more importantly, there was a patient that was homozygous loss of function for the gene called ATGL. And you'll be hearing about this later. ATGL is adipose triglyceride lipase. And when there's a loss of function mutation of ATGL, this patient had lipotoxicity of the heart. And upon biopsy, you can see that the entire vessel is filled with lipids. So what this means is that if you block the ability to break down lipids in the blood vessel, not necessarily in the blood, you can accumulate neutral lipid depicted by the red, showing that you have lipid accumulation throughout the vessel wall. So what do we know about how lipids accumulate in cells? There's been a lot of work in the past 10 years or so understanding the formation of lipid droplets in all cells going from yeast to man. And so when you have a chylomicron or VLDL-derived fatty acid, or just a pure fatty acid, that will be taken up by the cells, there then is an enzyme that will generate acyl-CoA species of fatty acids, and that acyl-CoA will then be processed by a series of enzymes with glycerol 3-phosphate as the backbone to form either diacylglycerols of phosphatidic acid and DAG, and then diacylglycerol in the presence of, again, fatty acyl-CoA in the presence of the enzyme DGAT will allow for the esterification of a neutral lipid into a lipid droplet core. So it seems that cells will not just allow free fatty acid to hang out in the cell, because free fatty acids can indeed be toxic to cells. So nature has designed a way of neutralizing lipids through the endogenous formation of lipid droplets. Conversely, there's a lipolytic pathway. So once you have triglyceride-rich triglycerides in a lipid droplet, there's a canonical pathway of lipolysis. The triglyceride can be first metabolized by the enzyme ATGL, depicted in red here. And there's an essential coactivator called CGI58 that interacts with ATGL, allowing for it to process the triglyceride. You then have a diacylglycerol that's formed. And there's a second enzyme called hormone-sensitive lipase that we've all learned about. This is the lipase that's regulated by catecholamines, the cyclic AMP. And you can see phosphorylation of HSL gives you the liberation of a second fatty acid moiety. Then there's a third enzyme called MAG lipase, and MAG lipase is not recruited to the droplet, but will metabolize the final fatty acid to form 3 fatty acids and glycerol. And then we know that these building blocks are used for energy, phospholipid biosynthesis, as well as for lipid metabolites. So when we first started doing these studies, we wanted to ask the question, do endothelial cells or do animals actually take up fat and store it in the endothelium? So Andrew Kuo in the lab and Monica Lee got together and they designed this following experiment. So we took a mouse that was fasted overnight, we gave it an olive oil gavage, and then we looked at the induction of triglyceride-rich lipoproteins in blood, and we also isolated the thoracic aorta to image lipid droplets. And this is a time course of triglyceride accumulation. You can see at time 0, they're very low. But after an olive oil gavage, by 90 minutes, you get a peak in triglycerides in blood that then wanes after about 180 minutes. If you then take the aorta and you fillet it open and you image lipid droplets using a Bodipi dye, or you can use oil red O, versus endothelial markers, and this is particularly looking at PCAM1 labeling, you can see that at 180 minutes, which is about 90 minutes past the peak in triglycerides in blood, you can see the accumulation of these neutral lipids in the endothelium by confocal microscopy. This is a higher mag picture of what the droplet looks like in the endothelium. If you quantify droplets from multiple experimental animals, what you can see is that there's an increase in LD numbers as a function of time that, again, get metabolized over another 90 minutes. So this really got us excited about the possibility that when free fatty acids are in blood, that those fatty acids are taken up by large vessel endothelium and stored into lipid droplets. So then we wanted to get more mechanistic, so we moved to in vitro and ex vivo experiments. So one way to form lipid droplets in endothelial cells in culture is using the free fatty acid oleic acid. Oleic acid is a monounsaturated fatty acid, and it's used in the field historically because it readily gets esterified into LDs. What you can see that there's a concentration-dependent accumulation of Bodipi positive neutral lipid in endothelial cells as you increase OA concentration. Also, there is lipolysis taking place. So this was an experiment where we took human endothelial cells, we loaded them with oleic acid overnight, then we just withdrew the oleic acid over 24 to 48 hours and looked at the decrease in BODIPI positive LDs. And what you can see over the course of 24, 48 hours, you get almost complete metabolism of the lipids. And if you put an inhibitor of the enzyme ATGL, or adipose triglyceride lipase again, you can see that you attenuate the reduction in lipolysis. And you can also measure triacylglycerol or triglycerides directly using a kit, basically. Then you can see that in normal endothelial cells, if you remove oleic acid, you get a time-dependent reduction in triglycerides. And this is antagonized by the inhibitor ATGL-statin. We also used a series of other inhibitors of the pathway, again, doing the same experiment. So you load with oleic acid, then you measure the release of free fatty acid or glycerol in the media. In doing this type of an experiment, you can either block ATGL, HSL, or MAG lipase using these different chemical inhibitors. And what you can appreciate is that, again, ECs will release free fatty acid, and this can be reduced by any of the inhibitors of the pathway. Another interesting approach to this was really a collaboration between Monica Lee and Andrew Kuo, is that they came up with an ex vivo system, which we thought would be much easier to do on a day-to-day basis. So what you do here is you take the vessels out of the animals, you clean off the fat, you mount them onto a cover slip, and then you can actually load with oleic acid overnight or at any time point you want to pick. Then you can look for lipid droplets using the BODIPI dye, or use your favorite endothelial cell marker. This case it's PCAM, and you can image using on-phos microscopy the amount of LDs in this vessel ex vivo. Now, if you do this experiment under control conditions, you can see a lot of LDs that are forming in the endothelium. And if you treat the vessel with a DGAT1 inhibitor, so DGAT1 again is a rate limiting enzyme for the formation of lipid droplets, you can completely aggregate the amount of LDs that you can measure. And if you do the same experiment, not with an inhibitor of synthesis, but an inhibitor of lipolysis, you can augment the number and size of the droplets. You can quantify as seen on the right over here. So with these three different approaches, we then wanted to ask some questions about the role of lipid droplets in endothelium. Another thing that we did was, in the original experiments, we used oleic acid. In vivo, it's more complicated because you have triglycerides that are already in lipoproteins versus free fatty acids that get taken up and esterified into triglyceride. So this is an experiment comparing oleic acid directly in endothelial cells versus chylomicrons or VLDL particles in the presence of added lipoprotein lipase. And the reason we had to add lipoprotein lipase is because endothelial cells do not make it. So if you put just chylomicrons in or VLDL particles in the absence of LPL, you get very little fatty acid released. So you have to actually put the LPL in to actually get mobilization of the triglyceride. So anyway, no matter how you look at it, it appears that you can get lipid droplets formed. And the nature of the lipid droplets look very different when you use OA versus chylomicrons or VLDL particles. Okay, so the question that we had was, what are the potential functions of LDs in endothelium? Why do you have to do this? What's the difference, and what can possibly be happening here? So we thought about three possibilities. One, perhaps the lipid droplet would protect cells from lipotoxicity. Perhaps when you have esterification of fatty acids into lipid droplets, maybe that fatty acid could be used for mitochondrial energy production. And thirdly, is it possible that fatty acids derived from droplets can be released from the cells and used by parenchymal cells, whether it be a skeletal muscle cell or a cardiac cell, for energy? So we examined these in three different in vitro systems. So the first thing we did, or Andrew did, is he looked at toxicity associated with OA, which is 18.1, palmitic acid 16.0, or stearic acid 18.0. And if you look at just lipid droplet uptake, what you can see is if you have no fatty acids, you don't detect them. But if you put palmitic acid or stearic acid at 1 millimolar concentration, you see very few lipid droplets that take place in the cell, whereas oleic acid gets rapidly taken up and esterified into LDs. So this tells you right away that there's a preference for monounsaturated fatty acids versus saturated fatty acids in terms of esterification. But if you take a half a millimolar of each, you can then start thinking about the possibility that maybe the OA was then pulling in the palmitic acid to esterify it into droplets. So to look at this in the context of ER stress, Andrew did Western blots for markers of ER stress, CHOP and BIP. So if you look at control cells, control endothelial cells, you can see without loading, there's very little CHOP induction, and there's basal levels of BIP. We also did this in the absence and presence of a DGAT1 inhibitor. And you can see the DGAT1 inhibitor had no effect, right? But if you load with oleic acid, you can see oleic acid by itself did not induce ER stress. But if you block DGAT, which means you block the incorporation of the fatty acid into a droplet, you get marked increases in ER stress responses. If you just give palmitic acid by itself, it is sufficient to induce ER stress based on CHOP and BIP induction, and the presence of a DGAT inhibitor does not make it any worse, showing that this is a lipotoxicity associated with palmitic acid. But if you mix oleic acid with palmitic acid, you can neutralize the toxicity in terms of CHOP induction in the presence of oleic acid. So that is similar to what Gene Schaefer published years ago, showing that oleic acid will drag palmitate into esterified reactions through DGAT. And we know it's DGAT dependent because if you put a DGAT inhibitor again, you reverse the protection that you see. And the same thing occurs with steric acid. And then if you do oleic acid in the presence of steric acid, you could again rescue the toxicity that's associated with steric acid. So this is sort of evidence suggesting that the presence of lipid droplets will reduce fatty acid toxicity. Next, we looked at energy production, oxygen consumption rate using seahorse experiments. And so these experiments, you can measure basal mitochondrial respiration, ATP production, and maximal respiration. And so when you load endothelial cells with oleic acid depicted by blue, there's really no effect on oxygen consumption rates. But we thought there might be a dynamic here because if you use a toxamir, which blocks CPT1, which is the rate-limiting enzyme or rate-limiting transporter that actually takes acylcarnitine into the mitochondria, you can see you reduce oxygen consumption rate if you block CPT1, suggesting that some of the fatty acid does get in to influence mitochondrial energy consumption or generation. And you can see if you look at maximum respiration with the presence of the toxamir and the presence of oleic acid, you see a very similar trend. If you look at glycolysis, what you can see is that when you load with oleic acid, you reduce glycolytic capacity depicted by blue, and that can be reversed partially again by a toxamir. And so this is considered what's called the Randall cycle, where when you have a reduction in glycolysis, you increase fatty acid oxidation and vice versa. And when you look at the maximum capacity as well as the glycolytic reserve capacity, there's really no effect of loading in the absence and presence of a toxamir. And the third thing we looked at was the release of free fatty acids. So the idea was when you have a triglyceride-derived fatty acid or just a free fatty acid in plasma, will that be esterified into a lipid droplet? And if it's in a droplet, can it then be released by metabolism to release the free fatty acid and glycerol that can possibly go into the underlying parenchyma? So what we did here is that we loaded ECs with oleic acid, we cultured them onto transwells, then did time-dependent accumulation and release of free fatty acid. And so this is an experiment where you can sample the fatty acid on the apical or basolateral side of the transwell. And what you can see after loading and release from loading, that there's a time-dependent release of free fatty acid after 6 and 24 hours in this transwell system. And if you put an ATGL inhibitor to block lipolysis, you can reduce this. If you put an HSL inhibitor to block homosensitive lipase, you can reduce it. But if you activate lipolysis with forskolin, which is a known activator of the PK protein kinase A pathway, you can increase the release. And the interesting thing to us at the time, we were hoping to see some polarization of free fatty acid release, but there really wasn't. It seemed to go in both directions in the absence of a cell on the bottom, which would be the cell that would take up the fatty acid. But if you do the same experiment in the presence of co-cultured myocytes or C2C12 cells, you can see after 6 and 24 hours, there's a little bit released into the apical side, but you cannot detect no fatty acid in the bottom side. It's all being taken up by the cells on the bottom and is esterified into droplets. So this really gave us the idea that the droplet-derived fatty acid could be released to the underlying parenchyma. So to summarize this first part of my talk, the idea is that free fatty acids or fatty acids derived from lipoprotein metabolism can be readily taken up by the endoplasmic reticulum, processed through a series of enzymes to generate lipid droplets. And then once in a droplet, there was a metabolism of the droplet-derived fatty acids through ATGL, HSL, and MAG lipase, releasing the free fatty acid that can be either protecting from lipotoxicity, provide fatty acid for fatty acid oxidation, or the release of free fatty acid when needed. Okay. So this also made us think about some other experiments that we did years ago. So when Carlos Fernandez was in the lab a while ago, he had this really interesting observation that if you have ApoE knockout animals that are bred to caveolin knockout animals, that you get a marked reduction in atherosclerosis using a variety of methods. And this image above just shows you how clean these vessels are from the double knockout animal compared to ApoE knockout alone. We also generated at that time an endothelial-specific reconstitute animal where we put caveolin-1 back into the endothelium of this double knockout animal. And what you can see is that there's a rescue of the phenotype, so you get more atherosclerosis. Now, the interesting part about this animal model is that it's very unusual, is that when you have decreased atherosclerosis in these animals, there's very high cholesterol and triglycerides in plasma. So the loss of caveolin actually increases plasma lipids, but reduces atherosclerosis, presenting a paradox. How can this possibly be where you have more lipids in blood, but less athero? And there's very few examples of this. Also at the time, we were able to show that the loss of caveolin also reduced LDL uptake into the endothelium, which may have a protective effect on the endothelium. But we became interested in this idea that perhaps the caveolin knockout mouse can metabolize triglycerides differently. The other thing that was interesting is that when you take endothelial cells in culture, you can purify lipid droplets away from other organelles. So this is a Western blot analysis of several lipid droplet markers, as well as ER markers, and then lipid raft markers like flotilin in endothelial cells in the absence and presence of oleic acid. And if you isolate lipid droplets, which actually float on this gradient, you can see enrichment of perilipin-2, perilipin-3, ATGL, the activator CGI58, and also caveolin-1. And we know it was a good preparation because if you look at lipid raft markers, flotilin, this was not enriched, nor was GRP94, a resident ER protein, or HSP90, suggesting that the droplets were highly enriched. Also, if you take caveolin-1 as an M-cherry construct, transfect it into endothelial cells, and give oleic acid, you can see that caveolin-1 can decorate the coat of the lipid droplet, suggesting that caveolin-1 plays a critical role in influencing lipid droplet formation. So what Andrew did at the time with Monica is we took wild-type animals, caveolin knockout animals, and caveolin knockout animals that were reconstituted in the endothelium transgenically, and we looked at LD formation after oleic acid loading. And what you can see is that you get nice loading that was diminished in the cav knockout, but then reconstituted in the reconstituted mouse. And this is the quantitative data looking at Bodipy intensity across several areas of the vessel. We also look at this in cell culture. So these are cells isolated from wild-type animals, caveolin knockout and caveolin knockout reconstitutes, and these are endothelial cells isolated from these animals. And what you can see is that you get marked accumulation of LDs, and this is reduced in the cav knockout ECs, and again, present when you reconstitute caveolin-1. This is the quantitative data on the right, looking at triglyceride content quantitatively. So then the question is, well, how does this occur? Does the loss of caveolin-1 influence fatty acid uptake, fatty acid synthesis, or lipid droplet lipolysis? So Andrew went through a series of experiments to try to test these three possibilities. So the first thing we did was look at fatty acid uptake mechanisms, because there was evidence that CD36 was regulated through caveolin-1. The problem with this idea was CD36, is really not really expressed in large blood vessels, number one. And secondly, it's not really clear what other transporters are important for fatty acid uptake. So we went to look at this using radioactive oleic acid. So this is the uptake of radioactive oleic acid at various time points in these three different lines of endothelial cells. And what you can appreciate is that there's really no difference in fatty acid uptake in these three different lines, arguing that the loss of caveolin is not influencing uptake. When you look at CD36 levels, it looks like it's a little bit higher in the endothelium of the knockouts. There's no effect on FATP4, which is another fatty acid transporter, which suggests that the uptake mechanisms were not really different between the three different strains. We then looked at fatty acid synthesis, and this is done using, again, radioactive oleic acid and glycerol. So triglycerides were measured using a TLC chromatography. And you can see here, this is the time-dependent formation of triglycerides in wild type, Cav knockout, and Cav knockout reconstitute. This is the quantitative data showing that the time-dependent formation of triglycerides was not affected by the absence of caveolin-1. We also collaborated with Richard Gross's group at WashU, and Richard did mass spectrometry to not only look at fatty acids, but fatty acyl composition of the different chains. And what you can see here, and this is wild type animals, Cav knockout and Cav knockout reconstitutes, in the absence and presence of oleic acid loading. What you can see is in the absence of OA, the amount of fatty acyl-CoA species is very low, but then when you load with oleic acid, it goes into the range of hundreds. And what you can see is what we saw by just measuring triglycerides directly. There's less in the caveolin knockout compared to wild type or reconstitute. When you normalize for the percentage of total triglycerides in the cell, you can see that there's no difference between wild type, Cav knockout, and Cav knockout reconstitute. Again, using a different approach of measuring tags, again, proving that the loss of Cav has really no effect on the synthesis of triacylglycerol. So then we looked at lipolysis, and these were actually pretty good experiments. It was very exciting to find this out. So what Andrew did is he loaded cells with oleic acid overnight and looked at the time-dependent release of fatty acid as well as glycerol formation. And what you can see is in the Cav knockout cells, you got nice release of both fatty acid and glycerol. And this was reverted when you put the caveolin gene back into the endothelium, suggesting that the loss of Cav increases lipolysis. We also looked at, this is just looking at the same data, but just plotting glycerol formation as a delta, and you can see that glycerol formation was increased in Cav knockouts. And this effect was blocked by ATG-L-statin, which normalized the data, suggesting that the blocking of the lipase was able to normalize the difference between the three different lines. We also looked at triglyceride formation over time in the presence of ATG-L-statin. What you can see is that in control cells, you get less triglycerides. Sorry, in caveolin knockout cells, you get less triglycerides. But if you block lipolysis, it almost normalizes the amount of TGs in all three cell lines. So the question was, what was the mechanism? And so this became the tricky part of the experiment. And I'm not really sure we have the final answer to this. But there was data from back years ago suggesting that caveolin-1 could regulate protein kinase A and cyclic AMP. So we measured cyclic AMP production as a function of OA treatment. And what you can see is in the Cav knockout cells, there was higher levels of cyclic AMP being generated after OA feeding of the cells. And this was again reversed when you reconstituted caveolin-1 knockout with caveolin-1. And when you look at pKa-dependent substrates, which is downstream of cyclic AMP, just by using an antibody that recognizes all the phosphoproteins that are pKa-dependent, you can see there's many, many more phosphoproteins in caveolin knockout endothelial cells at three different time points relative to wild type and to wild type knockout reconstituted. So then we had a situation where we had to try to examine how cyclic AMP or PKA can mediate lipolysis in the caveolin knockout animals. So one way of doing this was by blocking PKA with a drug that we use, H89, but there are several PKA inhibitors that work similarly. So when you look at, again, glycerol release from these cells, you can see that with forskolin to activate adenylate cyclase, you can get the release of more glycerol from the cells as a function of time, suggesting that the PK pathway, the basal PK pathway, can be activated even further with forskolin treatment. And when you look at forskolin-induced glycerol release, you can see that that was abrogated largely with H89, which is the PKA inhibitor. And when you do the experiment, just looking at triglycerides again, you can see that there's less triglycerides in the caveolin knockout cells. And this could be rescued to a large extent by using H89 to block PKA, suggesting that the enhanced PKA pathway was contributing to the increased lipolysis. And we're also able to show, and it was actually in the paper last year, is that this is associated with increased phosphorylation of hormone-sensitive lipase at the PKA site. Finally, we started thinking about, well, in caveolin knockout cells, how is PK activated? Is it just effects of caveolin 1 on either the cyclase or on PKA? Or is it possible that endothelial cells were producing a ligand to activate the PK pathway? And based on my past track record of working in the prostaglandin area, I started thinking about what prostaglandins activate the PKA pathway that are made by the endothelium, and the number one prostaglandin is prostacyclin. We know that arachidonic acid is a substrate metabolized by cyclooxygenase to form prostaglandin endoperoxides that's then metabolized by PGI2 synthase to generate prostacyclin. And the idea was there autocrine production of prostacyclin that was activating this pathway uniquely in the caveolin knockout system. So what Andrew did was the following experiment. He measured the accumulation of 6-keto-PGF1 alpha, which is the stable metabolite of prostacyclin in endothelial cells after OA loading in the absence and presence of a cyclooxygenase inhibitor, indomethacin. What you can see is that if you take endothelial cells, you load with oleic acid, you can see an increase in 6-keto formation in the CAV knockout cells, and this is blocked to a large extent with indomethacin. But when you load the oleic acid, you can see a marked increase in prostacyclin formation, again in an indomethacin-sensitive manner, suggesting that oleic acid loading leads to the formation of more prostacyclin. And that prostacyclin can then feedback activate PKA. And to look at the effect on triglycerides, we measured triglyceride content in the absence and presence of indomethacin, and we did get a partial rescue of the effect in the caveolin knockouts by indomethacin treatment, suggesting that this autocrine pathway is one potential pathway that can lead to the reduction of triglycerides in CAV knockout. It's not a complete rescue, suggesting that perhaps there's a direct caveolin effect also. So to summarize, we think that the autocrine production of prostacyclin can allow for activation of the prostacyclin receptor to increase PKA, leading to the increased lipolysis in caveolin knockout systems. And one question that we have, and we're going to do this using conditional caveolin knockout mice, is can the enhanced lipolysis that we see in the endothelium of CAV knockout mice contribute to the reduction in atherosclerosis in these animals? And we're trying to do those experiments presently. Okay. So to summarize this part of my talk, I think I was able to convince you that lipid droplets are formed and they're dynamic in endothelial cells using a variety of systems. You can initially drive LD formation by oleic acid in cell culture or by giving animals oleic acid. There's other ways of doing this in animals, such as starvation of animals or by giving a high-fat diet, but OA is a very convenient way of doing this. I think our data is pretty compelling, showing that caveolin 1 can influence droplet formation by influencing lipolysis, not triglyceride synthesis. And again, as I said before, it's not clear how the loss of CAV enhances cyclic AMP-dependent signaling, and lipolysis, and this is definitely worth exploring, and we'd like to do this. One possibility, but it's a difficult experiment, is that maybe Cav1 directly regulates the components of the lipolytic pathway. And then the thing that's kind of exciting to us is that, is it possible that these lipases that are important for LD formation can serve as a lipase for arachidonic acid generating prostacyclin? And when we submitted our paper, or right after publishing our paper, there was a paper from a group in Europe showing that ATGL inhibitors can block prostacyclin formation. So it's highly likely that there's a pool of prostacyclin that's derived from lipid droplet, not necessarily from the plasma membrane pools of arachidonate. Okay, so that's largely published data. I want to tantalize you with some unpublished data now that is keeping us moving in this direction. So the question then comes, what happens if we reduce LD turnover in the endothelium in vivo? So the approach that we took is we've generated ATGL flox mice that are bred to CDH5 mice to generate ATGL EC knockout animals. And if you take endothelial cells from wild-type animals versus knockout animals, you can see that there's about a 90% reduction in the level of ATGL protein, no change in CGI58 or VEGFR2 as a positive control. So we've just begun to characterize these mice in a variety of different paradigms. So I'll just show you some preliminary data. When you take vessels from these animals and you do the lipid droplet formation, what you can see is that if you just take the vessel from the animal, in the absence of OA loading, you can see endogenous fatty acid droplets that are accumulating in the endothelium by en face imaging. But if you take these vessels and you load with oleic acid, you can see much more accumulation in the ATGL knockout animal versus control animals. So this shows you that both basal and OA stimulated LDs are actually greater in animals that lack the lipase ATGL in the endothelium. Hang on. Okay. We also isolated cells from these animals and did it in cell culture. So these are, again, wild type and endothelial specific knockout ATGL cells from the lung of these animals. And you can see in the absence of oleic acid loading, there's endogenous lipid droplets suggesting they can't metabolize the fat very well. But if you load with oleic acid, the cells get very big and they're stuffed, they look like adipocytes almost, not quite as large, but they do stuff themselves with fat and they can't metabolize it very well. If you measure tag content using a direct approach, you can see in the ATGL knockout endothelium, you have much more triacylglycerol in the cells. But when you load oleic acid, the amount you have basally is almost identical in the OA-loaded controls. And then when you OA-load ATGL knockouts, you basically double your fatty acid in triglyceride in the endothelium. What about function? So Anna Gamma's in the lab took these vessels from these animals and measured endothelium-dependent responses, looking at relaxation to acetylcholine. And what she was able to show was that ACH-induced relaxations were rightward shifted, suggesting an endothelium-dependent relaxations that are largely nitric oxide dependent. If you look at phenylephrine as a vasoconstrictor, you can see that the curve is leftward shifted in the ATGL knockout, consistent with the loss of nitric oxide. And when you look at direct effects of a nitric oxide donor, such as sodium nitroprusside, there's no difference between the three strains, again implying that there's a selective reduction of nitric oxide dependent relaxation in these vessels. We then started examining eNOS levels in the vessels as well as in the cells. And this is an experiment where we did on-phos labeling of eNOS using an eNOS-specific antibody. And what you can see is in normal endothelial cells of these vessels, you see the tight perinuclear crescent of eNOS in these vessels. And you can see in the ATGL knockout, there's a marked reduction in eNOS protein levels by on-phos imaging. If you isolate the endothelial cells from the lungs of these animals, you can see there's a marked reduction in eNOS protein in the ATGL knockout endothelium, but no change in V, cadherin, or caveolin-1 levels. The mechanism of this is not clear. There's two hypotheses we're testing. One is that the loss of ATGL is blocking the formation of a PPAR ligand, which activates PPARs and PPARs then can activate eNOS transcription. The other possibility is that there's a degradative pathway independent of the mRNA that allows for the degradation of eNOS through either proteasome or autophagy-based mechanisms. The other thing that we started looking at was the presence of neutral lipid in other tissues from these knockout animals. So these are just cross sections of the heart, the liver, and skeletal muscle from ATGL knockout mice. And what you can appreciate when you look at the BODIPI positive staining is that you can see that there's areas of BODIPI positive endothelial cells that are accumulating fat in the heart as well as in the liver and in the skeletal muscle, suggesting that this is not just taking place in large vessel endothelium, but also in the microcirculation. So we're very much excited to test the possibility that perhaps ATGL knockout mice in the endothelium will have ectopic fat deposition into certain tissues that may trigger insulin resistant states. So we're presently doing experiments in high fat fed mice. And this is sort of a closer mag of the imaging from the heart and from the liver. And so we think this all kind of makes sense because there was a recent paper on cell metabolism by Peter Carmelite's group. And we think what's happening is that you can have free fatty acid or LD-derived fatty acids that participate in oxidative phosphorylation by influencing fatty acid oxidation. And we knew from this paper that if you knock out the gene CPT1, that you have a reduction of fatty acid oxidation resulting in endothelial dysfunction. So we also believe that if you block fatty acid mobilization by blocking lipid droplet-derived fatty acids, this would also trigger endothelial dysfunction and reduce eNOS activity. And we know that eNOS is also a major NADPH requiring enzyme, which is also down in animals that lack CPT1. So we think coming at it from a fatty acid pool pathway versus the actual mitochondrial effect here is another way of looking at the role of lipid droplets in influencing endothelial cell function. So our model is as follows. So if you have a chylomicron, a VLDL particle, or a free fatty acid, that DGAT will take that fatty acid from metabolism, convert it into a lipid droplet, and through metabolism by ATGL, you can release free fatty acids and glycerol. We believe that if you use ATGL-statin, or if you take ATGL knockout animals, by blocking this pathway, you get an accumulation of lipid droplets, you have a decrease in fatty acid oxidation, which leads to endothelial dysfunction. And the cool thing about this idea is that it would really be a link between fats in blood and endothelial dysfunction prior to any overt disease. So the animals are not atherosclerotic yet, they're not hypertensive yet, right? But they do have this early endothelial dysfunction that might be sort of setting the stage for things to get worse as you think about a second insult. Okay, so to conclude, we believe ECs really dynamically regulate LD turnover. Inhibition of triglyceride turnover in endothelial cells regulates fatty acid release, vascular function, and homeostasis, suggesting that defective triglyceride metabolism promotes EC dysfunction. And then the bigger questions that we're working on now is, can a reduction of ATGL activity trigger insulin resistance, impact cardiac function, with a response to exercise training where you need to have free fatty acids for mitochondrial respiration? And I think that the model that we're thinking about is that it's possible that endothelial cells have two pools of fatty acids for energy. The direct transfer mechanism that was initially elucidated from the earlier work I talked about, but we think there's a secondary step by which some of the fatty acids can be esterified into an LD as a secondary form of fatty acid. So I'd like to just, again, thank the people that did all the hard work. I'd like to especially thank Andrew Kuo, who initiated this project. And I'd like to thank Nabil Boutaghi, Ana Gamez, Alan Hay, and Monica Lee, who just moved to UIC about 2 months ago, who actually participated a lot in this project. I'd like to thank my collaborators, Richard Gross for the proteomics, Carlos Fernandez at Yale for the athero studies that we're doing, and Zoltan Rani, who we're collaborating with on some of this ATGL project. Thank you very much for your attention. Any questions? Hello?
Hello? Hello, this is Ana Gomez. I am a postdoc in Sessa Lab working in this project. And if you have any questions, can you please just send it? And I can read some of them I have so far. So the first question is, what is the mechanism involved in the transport of fats from endothelial cells to parenchymal cells?
That is the question. We don't know. That's a great question. Some of Stephen Young's work, where they actually measured a deuterated fatty acid being mobilized from a lipoprotein across blood into the endothelium and into tissue, suggests a very rapid transfer within seconds or within minutes of hydrolysis by lipoprotein lipase. The model was thought to be vesicle independent in just the hydrophobic nature of fatty acid getting across the endothelium in a directional way. But no one's really looked at transporters for the basolateral movement of fatty acids out of the endothelium. It's possible that there are fatty acid transporters that mediate the basolateral release of fatty acids. I'm just not aware of any of them yet. We do know that CD36 on the apical side is critical for uptake, though, of the fatty acid, but I'm not sure about the pathways for liberation on the basolateral side.
Okay, thank you. We have another question. Vascular triglyceride hydrolysis depends on lipoprotein lipase and hepatic triglyceride lipase. Do you have any data about LPL activity in your model?
We have not looked at LPL activity in what model in particular? In the wild type animal or in the ATGL knockout animal?
The question is just in your model, maybe cells or mice?
Yes. So in cell culture, there's no evidence of LPL being present in endothelial cells. It usually comes from it gets secreted by adjacent cells. In terms of in the ATGL knockout animal, we have some preliminary data suggesting that LPL activity is not different between the strains. I do not know if LPL is required for the formation of LDs in endothelial cells. If LPL is required, then you would predict with lipoproteins or with free fatty acids, you may not see as many droplets. But if you do see droplets in the absence of LPL, it suggests that perhaps the lipoproteins can also get into the endothelium in the absence of being esterified into TGs.
Okay, and there is the last question. In most immune cells, ATGL activity supports pro-inflammatory responses,
Yep.
but in non-immune cells like adipocytes or myocytes, the ATGL hinders the production of pro-inflammatory cytokines. Do you think that in endothelial cells, the absence of ATGL could be pro-inflammatory? Do you have any evidence in endothelial cells?
Yeah, that's a great question, and this is something that Anna's working on. And there was one paper suggesting that reduced ATGL in endothelial cells enhances TNF induction of adhesion molecules. So that would suggest that the loss of ATGL-dependent metabolism would promote inflammation. We're not necessarily seeing that in our hands, but to be honest with you, we haven't really explored it in a lot of detail. And the thing is, I think you have to think about what ATGL actually does. It's more, it is there for metabolizing the triglyceride. And if the triglyceride can't be metabolized, you might trigger other lipophagy pathways that then maybe perhaps link to other inflammatory pathways, perhaps through maybe the inflammasome. But we haven't really seen anything that's really jumped out at us. Especially in the context of in vivo systems, now that we know eNOS is down, it's possible that that also can contribute to any sort of pro-inflammatory state, at least in the context of the endothelial-specific knockout of ATGL. So this is a work in progress, and hope to be able to report to you within 6 months where we are with those experiments.
Oh, okay, I have another question. How caveolin 1 is regulated?
Yeah. So at the level of transcription, there's not a lot known. There was some evidence that it can be regulated by cholesterol, but that really hasn't made much of a splash. It doesn't seem to be highly robust from that perspective. In terms of caveolin regulation as at the protein level, there's evidence that it can be regulated through phosphorylation by SARC and SARC-like kinases, and that particularly tyrosine 14 can be phosphorylated, and that has been linked to endocytosis of caveolin and caveoli. Also, caveolin can be regulated through its interacting proteins such as the cavins, and so there are examples of caveolin being present in cells that don't have caveoli, but when the cells have caveolin-1 and other cavin molecules, you form the caveoli. So there's complex regulation through protein-protein interactions as well as through phosphorylation events on caveolin-1.
Okay, thanks. I think that's the last question. Okay, Linda?
No, you're not. Oh, hello? Hello?
Yes.
Yes, oh, can I ask a question?
Sure.
Sure.
Okay, so thank you for the nice talk anyway. My question is, you talked a lot about oleic acid and saturated fatty acids.
Yep.
I was just wondering whether this sort of effects can also be found when you use polyunsaturated fatty acids on them, specifically conjugated linoleic acid or just linoleic acid itself?
Yeah, that's a great question. There's lots of papers in the literature. We haven't done it in our lab yet, but it seems like PUFAs, particularly omega-3s, as well as linolenic acid, they tend to be incorporated more into phospholipids than they do into lipid droplets. And I don't know why that is. What's the mechanism of that? But to be honest, we haven't looked.
Okay.
We do see a lot of 24. We see a lot of arachidonate in the triglyceride of the droplet, but we really haven't looked for loading with like omega-3s or anything like that.
Okay. All right. Thank you.
Okay, so I think we're finished here. So, Linda, do you have anything to wrap up or?
I'm not sure what happened with Linda there, but we'd like to thank everybody for attending the webinar, and you'll be receiving a email in the for a survey, a follow-up survey. If you could fill that out, we'd appreciate it. And thank you very much from NAVBO.
Thank you.